have a much lower frequency. Find some in the ulcerative colitis patients. Now I said to myself, you know, this is a phony subdivision here. If you keep on making cuts, you'll find those granulomas. There are papers to this effect. The more blocks you cut and the more cuts you make, the more granulomas yet you find. So I don't think it's fair to describe this information as being 40% when in fact the total is 19%. And then there are some other things that are troubling. He says the labeling by in situ hybridization is on the myofibroblasts. Now class, you think the antigen is in the myofibroblasts? I don't think so. We're going to have to do some convincing, and his photographs are not convincing, to show me that the antigen is in the myofibroblasts. And he says that the macrophages are also positive. You can see them as brown spots. Well, let me tell you, from a pathologist's point of view, brown macrophages are sometimes troubling. And he finds no signals in the granulomas whatsoever. And that causes Seshi. Seshi also does some in situ labeling, but he does it on paraffin blocks as well. He finds 27 of 33 and none and none. But after he compares what he has found by his in situ labeling, and then he goes and he takes the whole tissue off a slide, take the whole slice off, and he does an attempt at IS 900, all of those were negative. This caused Rohal from the Netherlands to say that you have to interpret these staining properties in infectious diseases by the histopathologic content of the infected cells. The staining in the Seshi paper is a large rounded dot that exceeds the size of a single cell. Well, that was my thought when I saw these brown blots. It was like a Rorschach test. And then he goes on to say, and I agree with him, that given the high rate of in situ positives and the strong staining of signals, at least some of the IS 900 targeted PCRs should have been positive. Let me just see what the next slide is, because I'm being encouraged to stop. Well, here's another. I got to use one more story. Ryan from Ireland does laser capture. Now that's what we were all waiting for. Weren't we? We were all waiting for the laser capture. Go get those granulomas out of there and see if the granulomas are positive. And he used the PCR method for IS 900 on those laser dissected pieces taken from the paraffin sections. And he finds that in his 15 patients, 6 of them were positive. The granulomas were positive. Well, that's nice. You know, I'm happy for that. But let me tell you how he does this study. The first, he cuts 5 slides. They cut 5 slides. The first slide is an H&E so that they can find out where the granulomas are. Then they put that slide or either over the top or next to the second slide so they can microdissect the granulomas out, because you don't want them stained with the H&E. You just microdissect them out, and then they go into a vial, and then you do the PCR. And then the third slide is scraped off. The whole tissue is scraped off the slide, and that goes into a vial. So which of these is the correct information? Were 3 of his patients positive, or were 6 of his patients positive? More importantly, what happened to slides 4 and 5? If you got a disparity like this, don't you send the grad student back to the lab and say, redo the microdissection now on slide 4 and do the total PCR on slide 5? How about slides 7 and 8? You know, you got a whole block, you can keep on cutting, you can resolve this issue. This issue wasn't resolved. This is the way it was published, and it was said that when you do it on the whole tissue, it's a decreased sensitivity compared to the way that when you do it on granulomas. Well, I've shown you some examples of why I find fault with this information. Let me summarize. There are some questions about the validity of data. Just see if you agree with me now. There are some questions about the validity of data. For example, when someone reports on biopsies, that's quite different than a transmural cut. Have you seen the size of those biopsies? They're taken through an endoscope, and they're the size of the head of a pin, literally. That's a small sample. when those biopsies are taken, are they really sampling the disease or just surface organisms? Because the disease is in the submucosa, and those biopsy instruments only sample the mucosa. What was the bowel preparation before the biopsies were taken? We're not told that, not very well. How did they decontaminate the biopsy forceps between patients? There were a lot of issues a while back about decontaminating the endoscope, the colonoscopes between patients. They now expose them to, I think it's formaldehyde gas, but maybe they put them in a wet solution as well to disinfect them. Can some of the data that we're recovering here from biopsy samples be because of biopsy instruments that were never cleared of what was taken from the previous patient? And what about the water of the endoscope? When they put that endoscope in and they're searching around, there's a rinse water. You have two little buttons. One puts in air, one puts in water. What about that water that goes in at the time that the biopsy samples are being taken? I don't have answers for these. I'm raising questions. And then coming to those studies that were done where tissue sections were done, I would like to know, what did they do about the microtome knife? Did they use a separate microtome knife, a disposable microtome knife, every time that they cut these sections? Or did they wipe the microtome knife with alcohol or with chlorine? Sort of suspecting that they have disinfected the microtome knife. Microtome knives carry major tissues. Sometimes we find things like rumen microorganisms on a skin sample because the microtome knife has carried it. So that raises some issues. Did they, when they cut that slide with the microtome, they take a brush and they pick that ribbon of slide of tissues that have been cut. They pick it off with the brush. Do they change the brush every time? Or do you use the same brush? And then, if they're making slides, they pick that ribbon of cuts off and they put it on a water bath. A water bath, not the still water bath. They put it on a water bath. It gets changed once a day or twice a day. I'm merely pointing out for you some places where there may be some slippage here that may explain some of the discrepancies that we have seen. I've talked about them in the endoscopy suite and in the microtome suite, but they also happen in the lab. As you know, we're always very concerned. And in particular, I am told that in particular, when you get a mycobacterium in a lab, it's very, very difficult to get that out of there. To get it disinfected is one thing, but to get the DNA out of that laboratory is quite another. So that's something about the validity of the data. Lastly, let me ask you questions about the biologic feasibility. Do you really believe the milk of mothers with Crohn's disease contains Mycobacterium paratuberculosis? Do you really believe that ulcerative colitis patients are equally infected with Mycobacterium paratuberculosis? Ulcerative colitis is a disease that's different from Crohn's disease as BVD is from Johne's disease. Quite a different disease. Do you really believe that the PBMCs of pediatric patients with Crohn's disease have DNA, they're carrying around DNA of Mycobacterium paratuberculosis? What do you make of this disconnect between the data from the PBMCs and the data from the intestinal biopsies? Now, if you told me that a patient had a badly ulcerated, long segment of intestine, and that it had taken up some DNA from a mycobacterium, I might say, okay, that intestine might be positive, and maybe some of that would be some spillage that we found in the PBMCs, but I don't like this discrepancy that has been shown here. And do you believe that it's a highly resistant, Zeal-Nielsen negative form that's too small and underdeveloped to be seen by immunohistochemistry, that fails to induce a reliable serologic response, and that fails to respond to antibiotics that are directed against Mycobacterium paratuberculosis?